Here we'll learn the histology of skeletal muscle tissue. To orient ourselves, let's draw a limb in cross section. Draw the bone, then a few different muscle cross sections. Show that a single muscle may originate from more than one head, so we don't later confuse these with muscle fascicles. Now show that fascia envelops the muscles. Specify both the superficial and deep fascia. Show a representative neurovascular bundle within the deep fascia. As we'll see, these components enter the muscle. Then cover the limb in skin. Now let's address the muscle's organization. Show that epimysium, a type of dense, irregular connective tissue, envelops the muscle. Epi means upon and my means muscle. Next, show that perimysium divides the muscle into multiple wedges of muscle cells. Peri means around. Then draw a muscle cell within one of the fascicles. Show that endomysium surrounds it. Endo means within. Fill the fascicles with muscle cells and neurovascular structures. Let's review this on a histological slide. Show that epimysium envelops the muscle. Show that perimysium divides the muscle into fascicles. And show that endomysium lies within the muscle fascicle. It comprises a loose areolar connective tissue that maintains the extracellular environment for proper muscle cell functioning. Now draw a magnified view of a fascicle. Draw the muscle cells. Show that they're multinucleated. Each cell contains many nuclei. This reflects the process of muscle cell formation. Mature muscle cells form from fused myoblasts, embryonic cells. Each myoblast contributes its nucleus to the adult cell. Also show that each cell comprises numerous myofibrils. Show a representative myofibril, which each comprise numerous myofilaments, fewer thick myosin filaments, and more numerous thin actin filaments, which form a hexagonal shape around the thick filaments. Now let's look at a slide of a muscle fascicle. Encircle the muscle cell, which is covered in endomysium. Indicate one of the cell's many nuclei. Try to see the dot-like myofibrils within the muscle cell. Next, let's draw a muscle cell in a cylindrical three-dimensional view. Internal to the muscle cell, draw three representative myofibrils. Indicate that the muscle cell is bordered by the sarcolemma, its plasma membrane, and is filled with sarcoplasm, its cytoplasm. Show a muscle cell nucleus within the periphery of the muscle cell. And draw a representative mitochondrion. Muscle cells comprise numerous mitochondria. As we learn elsewhere, muscle fibers divide into three types. Type 1, which is slow and red. Type 2A, which is fast, intermediate. And type 2B, which is fast and white. These categories are based on their myoglobin content and contraction speed, and in related fashion, their number of mitochondria. External to the muscle cell, draw the external lamina. It's sometimes referred to as the basal lamina. Within it, draw a satellite cell, which are skeletal muscle stem cells, inactive myoblasts lying in wait. We think of them as the Army reserves. Upon muscle injury, they enter mitosis, fuse with other satellite cells to form a differentiated muscle cell. As you might predict, centrally located nuclei are a hallmark of regenerating muscle cells, whereas mature muscle cells contain peripherally located nuclei. Now along one myofibril, draw a web-like sarcoplasmic reticulum, which stores calcium and is a key component to coupling muscle cell excitation to myofibril contraction. Then draw bands of transversely oriented terminal cisternae of the sarcoplasmic reticulum. Show that they flank transverse tubules, T-tubules. They are tubular invaginations of sarcolemma. These structures are fundamental to excitation-contraction coupling. On the next myofibril, show its constituents. Indicate the A band, which refers to the length of the thick filaments. Think A for dark. They are anisotropic, or birefringent in polarized light. Show that the I band is the region along the thin filaments, between the thick filaments. Think I for light. They are isotropic. They do not alter polarized light. Now show that transverse bands, the Z disks, bisect the I bands. Indicate that a sarcomere, which is the contractile unit of the myofibril, comprises the area between the Z disks. Now on the next myofibril, once again draw the sarcoplasmic reticulum and terminal cisternae with intervening T tubules, and specify that they occur as triads at the AI junctions. Lastly, let's look at an electron micrograph of a muscle fiber. We see that the repeating light and dark bands of the sarcomere gives muscle fibers a striated appearance. First indicate the A band, remember A for dark, anisotropic. Then indicate the I band, remember I for light, isotropic. Show that Z disks bisect the I band. Show that M lines bisect A bands. They contain myomesin and creatine kinase. Indicate that a sarcomere is the unit that spans the area between adjacent Z disks. Finally, show the triad at the AI junction, which comprises T tubules and terminal cisternae of the sarcoplasmic reticulum. This concludes our diagram.